In today's video we will study about randomized clinical trial, sources of random numbers, purpose of randomization, data collection and documentation, masking or blinding, data analysis, phases in testing of new drugs, and ethical issues in randomized clinical trials. Randomization is the process by which allocation of subjects to treatment groups is done by chance, without the ability to predict who is in what group. A randomized clinical trial is a clinical trial in which participants are randomly assigned to separate groups that compare different treatments. Sources of random numbers are computers or calculators, which produces pseudorandom numbers based on a mathematical formula, or by websites such as random.org. True random numbers are based on true randomness outside of the computer, such as time to radioactive decay or atmospheric noise from radio. Primary purpose of randomization is to prevent bias in allocating subjects to treatment groups. Secondary purpose is to achieve comparability between the groups. Means there is no guarantee. Randomized trials are gold standard of study designs because the potential for bias is avoided. Stratified randomization is random assignment within groups defined by participant characteristics, such as age or disease severity, intended to ensure good balance of these factors across intervention groups. Data collection and documentation. Treatment has been assigned to the individual, and make sure that, if it's been received by the patient, later the outcomes are checked, that is beneficial or adverse effects of the treatment. Masking or blinding is used to increase the objectivity of the persons dealing with the randomized study. Subjects who can be masked or blinded are study participants, treaters, data collectors, data analysts, and investigators. Level of masking are non-blinded, single-blinded, double-blinded, or triple-blinded. A placebo is from Latin, which means, I will, please. Is a medical treatment such as operation, therapy, etc., which is administered as if it were a therapy, but which has no therapeutic value other than the placebo effect. A nocebo, which means, I will harm, is treatment like a placebo but which has a harmful result. Compliance is the willingness of the participants to carry out the procedures according to the established protocols. Dropouts are the participants who do not adhere to the experimental regimen during follow-up. Drop-ins are the participants who do not adhere to the control regimen during follow-up. There are two types of data analysis of randomized clinical trials. The primary analysis are pre-specified in the protocol, and the findings form the basis for recommendations and clinical decisions. They typically adhere to the intention to treat principle. Secondary analysis are supplemental and of various sorts. We encourage the use of the rich sources of data from large RCTs for these secondary purposes. Depending on the kinds of secondary analyses, whether they are pre-specified, and whether intention to treat analysis is used, the results range from being quite conclusive to being hypothesis generating. How to deal with non-compliance? There are two methods for this. First, monitor compliance, or observe it directly, counting the pills or conduct blood or urine tests. Secondly, use run-in periods. Run-in is a period before a clinical trial is commenced when no treatment is given. Example of run-in from the physician's health study is when 33,223 willing and eligible physicians were enrolled in a run-in phase during which all received active aspirin and placebo beta-carotene. After 18 weeks, participants were sent a questionnaire asking about their health status, side effects, compliance, and willingness to continue in the trial. A total of 11,152 changed their minds, reported a reason for exclusion, or did not reliably take the study pills. The remaining 22,071 physicians were then randomly assigned. Phases in testing of new drugs. In Phase 1 studies or clinical pharmacologic studies, the new drug is tested on a small group of people, ranges from 20 to 80 for the first time, to evaluate its safety. Level of toxicity is determined, along with metabolism, pharmacological effect, and safe dosage range, and identify side effects. In Phase 2 studies or efficacy studies, the drug or treatment is given to a larger group of people, ranges from 100 to 300 for efficacy, and to further evaluate its safety. In Phase 3 studies or effectiveness studies, the drug or treatment is given to a large group of people, ranges from 1,000 to 3,000, to confirm its effectiveness, compare it to commonly used treatments, and monitor side effects. In Phase 4 studies or post-marketing clinical trials, the drug or treatment is monitored to gather more information on risks, benefits, and optimal use. Common ethical issues in randomized clinical trials are to know. Is randomization ethical? Can the truly informed consent be obtained? When can placebo be used? Under what conditions should a randomized clinical trial be stopped earlier than originally planned? Thank you for watching, and don't forget to subscribe.